Today we finish off the respiratory system. We're going to talk about actually getting the gases into the blood and transporting them to the tissues. So we started off with the pulmonary ventilation, getting the air in and out. Then we talked about perfusion, getting the blood next to the alveoli so that the air and blood were close to each other. And now we're actually going to transport it across. We're going to take it to the tissues. We're going to move it into the tissues as necessary, bring back the carbon dioxide. Gas is always moved by simple diffusion. There's the cell membranes don't block gas movement because the molecules are so tiny. So if you have more molecules over here than you do over here, it's going to move across this way. That's just the way it is. And it will equilibrate relatively quickly if the barrier isn't too difficult for the gases to cross. So you have a diffusion pressure gradient. It's directly proportional to the molecules of gas you have, not how big the molecules are, but how many of them you have. You got 100, you got 50, you got a gradient going that way. So Dalton's law, we've talked about partial pressures a little bit in the previous lectures, just because, you have the partial pressure of 100 in the alveoli and 40 at the tissues. Okay, well, what exactly does that mean? Well, Dalton discovered that if you had a gas that had more than one gas in it, that the pressure exerted was proportional to the percentage of molecules that were a particular gas. In other words, if you have a balloon and there is 100 millimeters of mercury of pressure pushing against the sides of the balloon, and the gas inside is half oxygen and half hydrogen, 50 millimeters of mercury will be the oxygen and 50 will be the hydrogen. Because if the number of molecules is 50-50, exactly half of the partial pressure will be exerted by each. If you had if it was made up equally of oxygen, nitrogen, and hydrogen, then each of them would be pushing 33 1/3. So that would add up to your 100 millimeters of mercury. Now that's really handy because we don't have to come up with another calculation. If our atmosphere is 78% nitrogen, then 78% of our atmospheric pressure is nitrogen. And the rest of it is oxygen, because that's basically what our atmosphere is made out of. So the total pressure is simply going to be adding the pressure of this one, and this one to infinity, however many gases you have. So, knowing that, and that's useful, we talked about 1 atmosphere of pressure at sea level being 760 millimeters of mercury. So when we talk about just air pressure, if you're moving air in and out of your lungs, if your lungs are 761 and outside is 760, you have a pleural pressure of 1 higher than outside, and it'll blow some air out. If it's -1, it's -1 less than the outside, a little bit of air will come in. So we talked about that. But now we're going to talk about how much of that pressure is made up of the gases that make up our atmosphere. We're about 78% nitrogen, about 21% oxygen. It's not exactly that, but it's close enough for what we're doing here. Carbon dioxide is only about 0.03%. So physiologically, it's 0. Physiologically, there's no CO2 in the atmosphere. We make all the CO2 in our body through metabolism, and we blow out CO2, but the amount of CO2 that supports our trees and eats the sphinx away, that kind of stuff, is actually a very tiny amount. So it's only 1/4 of a millimeter of mercury. So for all practical purposes, we can say atmospheric CO2 is 0 for physiology. So really all we're worried about is 78 and 21 here. And if you want to know what the partial pressure in the atmosphere is of nitrogen, you multiply 0.78, 78% of 760, it's about 600 millimeters, and 0.21 or 21% of 760 is about 160. Add those together and you get 760. Okay, so that's what we were talking about. The percentage is, the partial pressure. Okay, so that's fine. The next thing that we find, which is also important to us, is that when you put a gas in contact with a liquid, the partial pressure of that gas will push X number of molecules into the liquid until the partial pressure in here is the same as it is out here. It will equilibrate. It doesn't mean it's the same number of molecules. It means that it is exerting that same force. So if you put oxygen in contact with blood, for instance, the oxygen will equilibrate until the partial pressures are the same. That's really handy too. You don't have to say it's 0.065 times or something like that. No. If the partial pressure of oxygen in your alveoli is 100, it'll be 100 in the blood too. That's really good to know. CO2 is a more diffusible molecule, so it'll take more molecules to produce that partial pressure, but still it will equilibrate too, so that you would have the same pressure inside and out. So whatever we put in the alveoli is going to transfer proportionally the same number into the blood. Very handy. So we know that we have, 160 millimeters of mercury of oxygen in the air. So we're breathing it in that. So why isn't the partial pressure in alveoli 160? That's because we're not getting all of it in there. We're mixing a lot of it with dead air that didn't leave the lungs the last time, right? You don't blow out every bit of air. Plus, when you pull air in, part of it stops in the dead space. So it's only the air that makes it into the alveoli to mix with that residual volume in there that is going to affect the partial pressure of gas in the lungs. And so as it moves into the airways, it mixes with old air. It gets fully saturated with water, which takes up a little bit more partial pressure space. And then it makes it into the alveoli and mixes there. By the time it gets from the atmosphere at 160 to the alveoli, it's about 100 in the alveoli. That's what it's supposed to be. Now, if you're breathing pretty fast and you have lots of oxygen around, it can go as high as 110. So that's not bad to have more oxygen in your alveoli, but 100 is the number you really need to remember. Remember, the big A, the partial pressure in the alveoli of oxygen, is about 100. Now, the partial pressure of CO2 tends to be about 40. Okay? Well, that's fine. Where is that coming from? It's not coming from the air. You aren't breathing in enough CO2 to make any difference. This is coming from your body. So the oxygen is going into the alveoli from your mouth, and the CO2 is going into the alveoli from your muscles, and they mix. You blow out the excess CO2. You keep taking in excess oxygen because you keep using it. You keep blowing off CO2 because you're making it, and you keep these numbers at rest right about here, about 100 millimeters of mercury of oxygen and 40 of CO2. That's what's the healthy level for you. Now, if we go to high elevation, things change. If you go to the top of Mount Everest, there's hardly any air up there. So the whole atmospheric pressure, instead of being 760, is about 250. It's only a third. Not enough for most people to be able to survive up there. Now, the fraction, which is oxygen, and the fraction, which is nitrogen, is still the same. It's still 78% nitrogen and 21% oxygen. But the number, it's 21% of not much. So the FO2 is the fraction. The fraction is always the same. It's 78% nitrogen, no matter what the atmospheric pressure is, and it's 21% oxygen, whether you have a whole lot of air or not much air because you're up high. Denver, it's certainly breathable there. In Taos, New Mexico, we were higher up than Denver. Our atmospheric pressure when I was living in Taos was about 600 millimeters of mercury. Well, 78% of that was nitrogen and 21%, still the FO2 is the same, the 21% was oxygen, but that meant we had a lot less oxygen up there. Before you got used to it, if you tried to go running, you'd be gasping for air in a couple hundred yards because there just didn't seem to be enough oxygen there. But the FO2 is the same, the fraction's the same, just the PO2, the partial pressure, which is 21% of that, is your partial pressure. Sea level, we know what it is. Now let's look at high altitude issues with oxygen. It's a real common problem for people, especially if you live at sea level. You go on a ski trip and you get altitude sickness. People go to Mount Everest and try to climb it and they die from altitude sickness. It's not exactly that they die from not having enough oxygen. They die from the way your body reacts to not having the right mixture or the right proportions of gases. So at high altitude, you have too little air, and therefore you have too little oxygen. The CO2 at high altitude is the same. It was already zero, right? Now it's even less. I mean, it was a tiny amount, now it's even tinier, but physiologically. So it's not the CO2 that changes at high altitude. It's the oxygen levels. So if you go to high altitude and your oxygen levels drop in your body, those blood vessels dilate to try to get more oxygen. In your brain, if those blood vessels dilate too much, you're in a hard case. There's no place for it to go. It creates pressure. It creates edema. There's too much blood pressure in your head. You can get swelling of the brain. You can get, first of all, you get headaches. Migraine headaches is one of the first things you get with high altitude sickness. But it can also cause cerebral edema and cause you to die if you went really high. But anyway, headaches, dilation. Pulmonary is exactly the opposite. When there's not enough oxygen in your pulmonary, in your alveoli, because there's not enough air, all the blood vessels around your alveoli constrict. They don't know that the rest of the lung also doesn't have oxygen. They're not smart. They're just used to constricting when there's not enough oxygen to make sure the oxygen goes somewhere else. The blood goes somewhere else. They all constrict at the same time. Your heart is pumping like mad to try to get enough oxygen to your body. It pushes so much blood into the lungs and it can't move through the lungs because it's constricted that you get pulmonary edema and your lungs fill up with fluid and you drown in your own juice. So this is what kills the majority of people who die on Everest if they don't fall off the mountain or something, is pulmonary edema. They get so they simply can't breathe and they die before they can get them back down to sea level. The best treatment for it is to get back down to sea level or put them in a hyperbaric chamber with excess oxygen or whatever to bring them back under control. Now, if you're breathing oxygen out of a bottle while you're up there, you're okay. Because if you have plenty of oxygen, your blood vessels react like normal. But you can't carry enough oxygen to breathe it 100% of the time all the way to the top of Mount Everest and back. So part of the time you're breathing oxygen, part of the time you're not. If you have plenty of Sherpas, they're stronger than you are. They can carry some more oxygen for you. But altitude sickness is a real issue for anyone who goes to high altitude. So here's a recap of your resting gas values. Atmosphere, oxygen's about 160 and CO2 is about 0. In the alveoli, it's about 100 for oxygen, 40 for CO2. Arterial blood, remember it drops 5 millimeters of mercury because of the bronchial circulation adding unoxygenated blood and a little bit of shunted blood at the bottom of the lung. those add together to give you not perfectly oxygenated blood. So your AA gradient is about 5 millimeters of mercury by the time you get to the arteries. The CO2 by then has equilibrated in the lungs. It diffuses very easily, so it equilibrates perfectly. And if it's 40 in the alveoli, it's 40 in the arteries. You go to the tissues. You give up oxygen to the tissues. That's why you went there. It comes back at about 40 millimeters of mercury. Remember that graph we had where it was 40, then it went up to 100, dropped to 95, it goes back down to 40, up to 100, down to 95, down to 40. Okay, that's typical of typical resting numbers. Your CO2, you pick up some CO2 from the tissues, so you leave your left side of your heart at 40. By the time you go through all the tissues and come back to the right side of your heart, you got about 46 millimeters of mercury of CO2. Now you'll give that off and get back down to 40 before you leave. Okay? Pick up PO2, pick up oxygen in the lungs, and you pick up CO2 in the tissues, and it all equilibrates and starts over. So now we have gotten the O2 and CO2 across the membrane into the blood. Just it's moving by its partial pressure gradient. Okay, there's more in the blood, so it moves to the alveoli. More oxygen in the alveoli, so it moves to the blood. How do we get so much to move? Well, the tremendous cross-sectional area of all those alveoli. You have a diffusion area the size of a tennis court. It's a big area for diffusion. It can happen within less than half a second as the blood passes through that little net of capillaries around the alveolus. Before it's even halfway past, it has equilibrated. And the rest of the time it's just going for a free ride in case it needs a little bit more something, but it equilibrates very quickly. Movement through capillaries is slow, so there's time. I mean, it takes a second for that tiny distance for it to go around a microscopic alveolus. About half of it equilibrates, the other half it's already equilibrated. The respiratory membrane we talked about being very thin, just a simple squamous cell on the alveolar side, simple squamous on the capillary side. And then the diffusion coefficient. Like I say, CO2 diffuses real easy, more easily than oxygen. So it's there. Oxygen takes half a second to diffuse all the way across. Respiratory membrane, we talked about. It has the type 1 epithelial squamous cells plus the 2 surfactant cells. It's very thin. We've seen this one before. You've got a cell, a basement membrane. Here's the capillary. Here's the capillary, and there's the alveolus. Here's your type 2 cells making surfactant. Now, your diffusing capacity can change a lot with disease. This is the same magnification, but this lung has emphysema. This is a nice dense sponge with lots and lots of alveoli, lots of tissue, lots of membranes, lots of blood vessels in here to cross over. Here, a lot of it is just absolutely dissolved away. Remember, with emphysema, you have too much air in there. The term emphysema actually means too much air. And pulmonary emphysema means there's too much air in your chest. It's in your lungs, but but it's not inside of lung tissue necessarily. It's in these big gaping holes where tissue used to be. So, with lung problems, the AA gradient becomes too large. You can get the air into the lungs, but you don't have enough healthy tissue to move it across into the blood. So again, you could have 100 in your lungs, but only have 70 in your blood if you can't get that oxygen across the membrane like you're supposed to. Now remember, your partial pressure when you reach the arterial blood is like 95 millimeters of mercury, which means that about 98% of the blood has been oxygenated, and it drops you down to 95 millimeters of mercury. It's too bad these numbers are so close to each other, it's easy to get them mixed up. But just remember your AA gradient is about 5 under healthy conditions. Now that is enough to oxygenate your hemoglobin on your blood, inside your red blood cells, almost completely. So you have your hemoglobin is oxygenated, and it's going to take that oxygen and deliver it to the tissues. 97%, there's another 90, but just say almost all of your oxygen is transported by a hemoglobin. Oxygen doesn't dissolve in plasma very well. So as soon as it dissolves, your hemoglobin pulls it out of solution so you can get some more oxygen in there and pull that out of solution. The hemoglobin loves oxygen at the lungs, and it'll stick on really fast. Pull it out of that solution so you can get more oxygen molecules into the solution. So virtually all of your oxygen has to be carried by hemoglobin. That's why if you don't have enough red blood cells, you die of hypoxia, because you can't just dissolve it in your plasma and do enough of a job. You have ferrous iron located in 4 locations on each hemoglobin molecule. These are the hemes. The hemes are reddish. It gives you your red appearance because of the iron. And so it's the iron that is binding the oxygen within the hemes of the hemoglobin. Here are the hemes, 1, 2, 3, 4. And they have the iron in the center. And you have hemoglobin. Okay, well, there are several different kinds of hemoglobin. There are only 3 that I need you to know for sure what they are and what they do. Excuse me. When you're a fetus, you have what's called fetal hemoglobin. It's a hemoglobin that has a stronger affinity for oxygen than mom's, because when mom's blood passes by the fetus in the placenta area, the baby's blood, which is kept separate from mom's but is coming right next to it, has to want that oxygen more than mom's, so it pulls the oxygen on to the fetal hemoglobin. So during fetalhood, you have nothing but fetal hemoglobin, and it is taking mom's oxygen as necessary. Red blood cells only live a couple of months, and shortly after you are born, they start dying off and you're making new ones, and the new ones you make have adult hemoglobin in them. Adult, that's the same as mom's. It has a normal affinity for oxygen that works really well at sea level and some distance above. And then sickle cell anemia has hemoglobin, HB, hemoglobin sickle. Here's hemoglobin fetal, hemoglobin adult, and hemoglobin sickle. And it's the hemoglobin itself that causes the change in shape of the red blood cell, that causes it to elongate and sickle and get hard so it won't fit through capillaries. So sickle cell disease or sickle cell anemia causes the sickling process of those red blood cells so they can't fit through capillaries, and you have all the way up to lethal blood flow problems. We have another hemoglobin type molecule called myoglobin. As you know, myo means muscle. So this is a hemoglobin type molecule inside skeletal muscle in particular. And it makes your skeletal muscle, especially the oxidative ones, really red looking, the red fibers. It binds hemoglobin, and it wants hemoglobin more than your adult hemoglobin too. So the myoglobin will, sort of like the fetus does, will pull that hemoglobin out of your blood and into the cell so you can use it there. So the more myoglobin you have, the better you can move oxygen into the cell from the oxygen that's in the hemoglobin and use it inside the cell. Okay, so if you're going to do this lecture, you've got to go through the oxygen hemoglobin dissociation curve, because this describes how oxygen sticks to hemoglobin in one place and lets go in another. It wouldn't do any good for oxygen just to stick to hemoglobin, always, because then it'd just go round and round and round. It would never go to your tissues. What you need is a molecule that will grab hemoglobin at the lungs and then let go at the tissues. And that's what this curve is. Under what conditions does it stick, and under what conditions does it let go? So the factors you find in the lungs increase the affinity of oxygen for hemoglobin so that it sticks really well. And the factors we find in the tissues decrease that affinity so it lets go. Affinity means I want it. So if you increase it at the lungs, you grab it. You decrease affinity in the tissues, you let go. Here we go. In the lungs, we have a partial pressure of oxygen very close to 100. Great. 100% of your hemoglobin has oxygen stuck onto it. How convenient that that should work out that way. So at the lungs, we're going to grab it. You get down to the tissues, and even at rest, you have 40 millimeters of mercury. Well, hemoglobin lets go of oxygen better. So you're letting go of this amount of oxygen just by going by the tissues. Then you come back and you grab it, and you go down and you let go. When you exercise and burn up oxygen really fast, you can lower your partial pressure down in the tissues to like 20%. Look how much more oxygen will let go during exercise. So it's still picking up all of it in the lungs during exercise because you're breathing really fast. You keep the oxygenation in your lungs fine by breathing fast. And yet, when you get to the tissues, you're letting go of a greater amount. So there's a more dramatic oxygen delivery during exercise than there is at rest, just as you would want it to be. Here's the curve for a couple different kinds of hemoglobin-type molecules. Here's normal human, mommy, hemoglobin. It's 100 at 100. And at 40, it's about 75%, and at 20, it's about 25%. So that's the normal curve for normal hemoglobin. Fetal hemoglobin likes it more. So every place along here, this hemoglobin is going to grab it more than this one does. It's pulling it off the baby. Pulling it to the baby from the mom. Myoglobin, same thing. If you get hemoglobin past a skeletal muscle, it's going to grab that oxygen off the hemoglobin because it has a higher affinity. And they put this in, it's interesting, llama. This is the beast of burden of South America. The 1L llama is a priest and the 2L llama is a beast. The llama developed up in the Andes where there's hardly any air. And so they developed a hemoglobin that, don't do that, they developed a hemoglobin that really, really likes oxygen. So even if there's only 40 or 50 millimeters of mercury of oxygen in their high mountain area, they'll still grab it really well. And they'll let go when it gets down to the tissues. But at a much lower partial pressure of oxygen out in their atmosphere, they will have more oxygen and be able to survive up there. Now you can alter this curve especially during exercise, through a couple of ways. I said everything you find in the lung makes it want to stick better. Everything you find in the tissues makes it want to let go. So these are the things that change from the lungs to the tissue. The first thing is you have more oxygen in the lungs than you do in the tissue, and when there's more oxygen, it sticks better. So that's good. That's a good overall one. Secondly, though, you have more CO2 in the tissues than you have in the lungs because you're blowing the CO2 out of the lungs and it's being made in the tissues. CO2 binds to hemoglobin too, and they don't like to be on the molecule at the same time. So if you go where there's a lot of CO2 and it binds, it pushes the oxygen off. Well, this is really helpful because you're at the tissues. You want the oxygen to leave. So you grab CO2, the oxygen leaves. So at the tissues where CO2 is high, the affinity for oxygen gets lower and you let go of the oxygen. pH, more acid, makes you let go of oxygen. Well, that's good because your tissues make acid. You blow off CO2 in your lungs, there's less acid there. The pH is higher. Acid is a low pH. So low pH at the tissues makes you let go of oxygen better. Temperature. High temperatures make oxygen let go easier. Well, that's good because your tissues make heat. So you go to the tissues, you find CO2, you find acid, lower pH, and you find higher temperatures, and all of those things help you let go of the oxygen at the tissue. This is a good thing. It makes hemoglobin work even better than it already was going to work. And especially during heavy exercise, you make more acid, you make more CO2, and you make it hotter. So during exercise, you let go even better. And yet there's one more thing that you can make. This is 2,3-bisphosphoglycerate, 2,3-BPG, is made as part of the glycolysis process, glycolytic pathway. You make 2,3-BPG as part of glycolysis. Well, glycolysis happens when you're anaerobic. So if you start working a muscle so hard that it's working anaerobically, it will make 2,3-BPG, and that makes the hemoglobin coming by let go better, so that tissue starts getting some oxygen. So every tissue that's working hard and needs more oxygen can ask for it by making 2,3-BPG just by the fact that it's working anaerobically. So CO2, acid, temperature, 2,3-BPG. Those things during exercise help you deliver the oxygen to the tissues that are working. This is it, 2,3-BPG. The product made in glycolysis is actually 1,3-BPG, but it converts to 2,3-BPG, and then you get a lot more oxygen. Great, great, great. So, if I were to look at, let's say this is a normal dissociation curve, and I'm saying, okay, well, what's going on here? What does the dissociation curve here? Well, this is probably during exercise. I have lowered this so it's letting go of, at every point along here, it has less oxygen stuck onto it. These are conditions found at the tissues. These are conditions felt more at the lungs. This is breathing fast. This is working hard. Okay? So the curve can be altered by those factors. And just know that if you're lowering your line here, it means that you're letting go of more oxygen. So you must be at the tissues. You must be in working tissues. Now, how does carbon monoxide affect oxygen carrying capacity? Well, as it turns out, carbon monoxide binds to hemoglobin just like oxygen does, but it binds too hard. It binds so hard that it won't let go, and now oxygen can't bind either. Once it's bound, it changes the conformation of the hemoglobin molecule, and you can't even bind any O2. So you've got perfectly good hemoglobin going round and round and round, but it's bound to carbon monoxide. It can't carry oxygen, and you die of lack of oxygen. You don't even notice that you're losing oxygen, because low oxygen in the plasma, you still have normal oxygen in your plasma, but it's not very much it's not enough to help. What you need is to be carrying a lot on your hemoglobin, and you're not. And so, in something like 7 hours or so, it can clear, but by then you've been dead for 5 hours, so it doesn't help very much. This is where hyperbaric oxygen, here's another treatment that actually does work, because you can deliver so much oxygen that the oxygen displaces the carbon monoxide. It wins, gets rid of the carbon monoxide. You can do it in about 20 minutes, and maybe you can keep the person alive that long. One problem is, like I say, your arterial sensors test the oxygen that's in your plasma. They can't test what's stuck to the hemoglobin. So they test your plasma, and you're breathing normally, so your plasma has normal oxygen in it. You just can't get any on your hemoglobin, so you're only getting 3% of your oxygen being delivered to your body, and you die. You don't even start breathing fast, because you have normal sensation that your sensors are telling you have the right amount of oxygen. So that's what's so scary about it. People are always just dying in their sleep. They don't wake up gasping for breath. They just die without ever regaining consciousness. Okay, so now how does this hemoglobin, how much oxygen does hemoglobin need to carry? And how much can it carry in your blood? Well, humans have about 15 grams of hemoglobin per deciliter of blood. A tenth of a liter of blood has about 15 grams of hemoglobin in it. And each gram can carry 1.34 mLs of oxygen, which means you're carrying about 20 milliliters of oxygen in every deciliter of blood. It's called 20 volumes percent. It's not that 20% of your blood is oxygen. It's just that every 20 volumes, every 100 volumes, 20 per 100, every 100 volumes of blood will carry 20 volumes of oxygen. Sounds confusing, but a deciliter, so 20 mLs per a deciliter is 100 mLs. That's why they do it per deciliter. So 100 mLs of blood has 20 mLs of oxygen. 100 quarts of blood would have 20 quarts. 100 gallons of blood would have 20 gallons of oxygen. That's what 20 volumes per 100 means. It's 20 per 100 of anything, any volume. So if you pump 5 liters of blood per minute, what's your total oxygen capacity per minute? Well, 5 liters, what's 20% of 5? 1, right? 20% of 5 is 1, So 1 liter of oxygen per minute is being delivered to your tissues if you're pumping 5 liters of blood. If it were 10 liters of blood per minute, if you're exercising, you'd be carrying 2 liters, right? Okay, because 2 is 20% of 10. Okay. Now, the other thing on this is important is that men have more hemoglobin than women. It's because their hematocrits are higher. They don't necessarily have more in each red blood cell, but they have more red blood cells. So every deciliter of blood for a male carries more hemoglobin than the female. 16 to 14. Averages out about 15. So males have an aerobic advantage for things like marathons and whatever. Every female marathon runner on the planet could beat me, and I'm a male, But with equal training and equal talent, the guy has an advantage. He can carry more oxygen. What about carbon dioxide transport? You're bringing it back from the tissues. Well, a little bit more can dissolve in the plasma. Remember I said that CO2 dissolves really easy and transports really easy and that kind of stuff. So you've got a little bit more in the plasma compared to oxygen, which is almost none. Hemoglobin will carry about 30% of it. Well, that's a chunk. You know, it displaces the oxygen, and hemoglobin carries the oxygen back. Carries the CO2 back, carries the oxygen there, carries the CO2 back. But here's the take home. More than 60% of your carbon dioxide is carried as a compound called bicarbonate. The bicarbonate ion. Carbon dioxide combines with water, and there is a enzyme called carbonic anhydrase that converts this and this to carbonic acid. It loses one hydrogen and becomes bicarbonate. Then if you need it to go the other way, this is a reversible reaction. The bicarbonate picks up that extra hydrogen again and converts back to CO2 and H2O, and you blow out the CO2 and you keep the water. Okay? It's a great way to carry CO2 away from your tissues. Your hemoglobin can't carry it all. So you combine a lot of it with water, you take it to the lungs, and then you split and blow out the CO2. Okay? The CO2 goes, and H2O, some of it goes out. You know, you breathe some atmospheric air out, some water out. But most of the water you keep, and you blow out the CO2. So here we have blood is going past the tissue, working tissue. CO2 comes out, a little bit binds, a little bit stays out here, a little bit dissolves in the plasma. Some of it binds to the CO2, but the majority of it combines with water using carbonic anhydrase to make carbonic acid, which becomes bicarbonate, loses the hydrogen, and floats around in your blood as bicarbonate. Bicarbonate is your buffer that you use to buffer acids. Because it's a negative ion, it can pick this hydrogen back up. If you start making too much hydrogen, you can pick it back up, run in this direction, and make water and CO2 again, which is what you do with the lungs. That's how you're getting rid of acid. You're taking the hydrogen back onto the bicarbonate, splitting and blowing out the CO2, and presto, the hydrogen is now gone, back as part of the water molecule again. Here's the other direction, okay? The bicarbonate comes in, picks up a hydrogen, you blow off the CO2, and the water just hangs around wherever it wants to. How do we control all this? How do we tell if we have too much oxygen, too little oxygen, too much CO2, too little CO2? We have respiratory centers in the brain stem that will sense how much of everything there is and make you breathe faster or slower. You know, they have sensors that are not part of the voluntary system. You can breathe faster and slower on purpose, too. Those are skeletal muscle we can control. But we have an involuntary control of these skeletal muscles that's located in the brainstem, so you don't die in your sleep because you forget to breathe. CO2 is the main driver of ventilation because this is what we make. So if we're making too much CO2 and not getting rid of it, we breathe faster. That's what mostly drives our ventilation. So when CO2, as you breathe faster, you blow off the CO2, right? Ventilation drops, CO2 starts building up. So you breathe faster, CO2 goes down again. It's an inverse relationship between how fast you're breathing and how much CO2 you're going to build up. Your respiratory centers are located in the brainstem. You have the pontine group, which is in the pons, not surprisingly. And then in the medulla, you have dorsal and ventral respiratory groups. And all of these have their little functions, which you don't have to memorize for this class. But, one makes you breathe deeper, one makes you breathe in more, one makes you breathe out more. They have different ways of controlling your breathing. So if you stimulate this guy, maybe you'll exhale more. You stimulate this guy, you inhale more. You stimulate this guy, maybe you'll breathe faster. So when you get the sensor, the signals coming in from your sensory mechanisms, it will stimulate these right groups, these correct groups of nerve cells here, and they'll send out the signals to the right inspiratory and expiratory muscles. You have a central pattern generator in there. It's kind of like your SA node on your heart. And the central pattern generator will keep you breathing even if you don't have stimulation going on much. It'll cause you to breathe in and breathe out at a normal pace, even if nothing else much is going on. But that central pattern generator, just like your SA node during exercise, can be stimulated and make your heart speed up. You can alter the central pattern generator's pattern so that you breathe faster and deeper. You have various chemoreceptors, sensing how much oxygen and CO2 is in the blood. Pulmonary stretch receptors, if you breathe in really deeply, you send a signal that inhibits further breathing in. Baroreceptors, if the blood pressure goes up, you assume you're exercising and therefore you start breathing faster. Irritant receptors make you cough. Proprioceptors, if I move my arms a lot, I'll start breathing faster even before I need the oxygen because I'm signaling my central pattern generator that I'm moving, I'm going to need more. Thermal receptors, if you get hot, you breathe faster. So all of those things send in signals to the central pattern generator, and it sends signals to these little respiratory groups and make you breathe in and breathe out. Here we go. Sensory input to the central pattern generator. Also, you can voluntarily control it from the cortex, so it can get signals from your cerebrum, from your cortex also. And then it sends the signals to the respiratory groups, which then causes you to breathe in and breathe out. Now, these chemoreceptors are designed to tell you if your CO2 mainly, because that's your main driver, but also O2, is okay. And if it's not, they can make you breathe faster or less. So we have central chemoreceptors in our cerebrospinal fluid to see if there's enough, if the gases are okay there. And you have arterial ones that are in your the aortic arch and the carotid sinus. These are in the main places where you're going to be sensing blood leaving your heart and tell you if it's got the right gas mix. And then if anything's off, they're going to send signals back to the central pattern generator and make you breathe differently. When you have too much CO2 or way too little oxygen, you do respond to oxygen too, but it's secondary to CO2. But if you have too much CO2 or way too little oxygen, you get this air hunger. You know what it is. If you try to hold your breath too long, you get air hunger and you just can't control it. You can't hold your breath till you pass out. It forces you to breathe again before you pass out. Central chemoreceptors don't test oxygen, they just test CO2. CO2 can cross. Carbonic anhydrase converts it to bicarbonate and hydrogen. You respond actually to the hydrogens that are being produced by the CO2, but it's a measure of your CO2 levels in your brain that would cause you to breathe faster. Central chemoreceptors only respond to central CO2 responds to central levels of CO2 to make you breathe faster or slower. Your peripheral chemoreceptors, located in the carotid, the bifurcation here, there are some in the aortic arch, but primarily these are located in the carotid sinus up here, where you get the split from the internal to external carotids. And these also mainly respond to CO2, but if oxygen gets low enough, they'll respond to that too. So peripheral can do both. First of all, CO2, but secondarily, PO2. That's how, if you go to high elevation, the CO2 doesn't change, but once the PO2 drops below a certain point, you start breathing faster. So you first get there and you think everything's okay, but you walk up one flight of steps and the oxygen's too low and you start huffing and puffing. And again, that's not because there's any more CO2 there. It's because the PO2 is too low, and that's being picked up by these peripheral chemoreceptors. Okay, that's a lot of stuff, but it's all good stuff.